Awesome. Okay. So, hi everyone. Today I would love to tell you about my research in tallgrass prairie, specifically a tallgrass prairie restoration experiment that's been going on at Konza Prairie Biological Station near Manhattan, Kansas for over 20 years now. And my presentation is titled Heterogeneity in Nitrogen Affects the Phenology of a Dominant Grass in Restored Prairie. That's a bit of a mouthful, but we will dig into what all that means. So first, as a quick introduction, as you may know already, the tallgrass prairie is a very diverse and very heterogeneous ecosystem. So what does that mean by when we say that this ecosystem is very heterogeneous? Well, it means that the ecosystem is characterized by this patchy distribution of soil nutrients and also of different species that are growing in the prairie. So as you can see just by looking at this picture of prairie, you can already see a couple of distinct patches. For example, a patch of yellow flowers growing right here, patch of these violet flowers growing right here, or you can also see patches, for example, a patch of upland versus a patch of lowland area. And then one thing that you're not seeing is patches of soil resources, such as nitrogen, or where water is being retained more in the soil. So it's a very complex and diverse ecosystem, and heterogeneity has been shown to be one of the explaining factors in that complexity and diversity. And you may be familiar with the importance of bison and fire in prairie. And this is one of the proposed reasons for why bison and fire are so important in a prairie, is because they create this patchy distribution of resources. For example, when bison poop, they leave a patch of nitrogen. So one of the challenges that we face when restoring prairie is that we're often restoring from agricultural land. And one of the reasons that's a challenge is because in the process of farming, soil is tilled, which homogenizes that once very patchy distribution of soil resources so that everything is very uniform. And one theory is that this is one of the reasons why it is so difficult to obtain the same levels of species diversity that we have in remnant untilled prairie when we're restoring prairie. And the logic behind this follows something called the environmental heterogeneity hypothesis, which is that variability in the environment increases niche availability to allow more species to coexist. So essentially the idea is that an environment with these distinct patches is going to support the growth of different species which prefer maybe one patch over another patch. So this led us to wonder if we could induce heterogeneity in order to have more diverse prairie when we are restoring from agricultural land. So we're asking, can we add nitrogen? Can we reduce nitrogen? Can we adjust soil depth? And can we adjust the amount of water that different patches of soil is receiving? And if we do that, how is that going to affect the dynamics of the prairie plants that we see? So this is the driving logic behind this experimental restoration that my research is taking place in. That was originally created by my advisor, Dr. Sarah Baer, around 20 years ago. And my research specifically is looking at one species, which is Andropogon girardii, or big bluestem grass. And big bluestem grass is really important to the other species and the overall species dynamics in prairie ecosystems because it's just so dominant. Big bluestem can make up more than 80% of the biomass of the prairie plant community. in a given year. So the way that Big Bluestem is using and allocating resources to produce this biomass has these important implications for how those resources remain available to other species throughout the growing season and from year to year within a prairie plant community. And one of the ways that we can study resources and development is from a temporal perspective. And when we're studying the temporal aspect of plant development, we're studying phenology, life cycle timing. It's essentially the study of how plants develop through time and the timing of important stages in plant life. And most commonly, we're looking at flowering, and grasses do flower as well. And flowering is especially an especially important turning point when studying phenology, because this marks the point at which plants switch from allocating their resources to growing bigger versus to reproducing and passing on their genes. So limited resources in this ecosystem necessitate a trade-off in how those resources are allocated. Plants can't constantly allocate resources to growing bigger without reducing the amount of resources that they can allocate to reproduction. Now, one thing about phenology is we do know that the abiotic environment, the soil resources, and the and other qualities of the environment, we know that these affect the phenology of species. It's been established that temperature, especially, and particularly warming, has this effect on the timing of plant species life cycles. And one way that you might even have noticed this in your own life is allergy seasons are becoming more extended, and that's because as the climate warms, flowering is happening earlier and earlier. And so that's kind of interesting. Other variables that affect phenology include atmospheric carbon dioxide, soil nitrogen, and precipitation, although those last two are have more variable effects depending on the species that you're looking at. And so those are of particular interest to my research. I wanted to see how those specifically affected Andropogon giardii, big bluestem. Another interesting thing is that the biotic environment, that is to say the biological aspects of the environment, such as species diversity, are also known to affect phenology. So species growing in less diverse assemblages have been shown to flower earlier than species growing in more diverse assemblages. And variation in the timing of flowering has also been shown to be affected by species diversity. So this points to a potential link between the abiotic environment, the biotic environment, and phenology. So we recall that the tallgrass prairie has this heterogeneity, and that in our experimental restoration, we're inducing heterogeneity to understand how that is important and might affect species dynamics. So we come to this question: how does spatial heterogeneity of soil resources affect the phenology of big bluestem? And then if we take a closer, more fine lens into this question, how specifically do nitrogen and drought or precipitation affect the phenology of big bluestem? So to answer this question, we're working in the Flint Hills at Konza Prairie Biological Station, which is in the tallgrass prairie ecosystem. Our restoration area is kind of a special little spot in Konza, since most of Konza is remnant tallgrass prairie. It's never been farmed, it's never been tilled, but our area is historically agricultural land. So we took this small plot of agricultural land and we're using it for this restoration. the design of our restoration follows a block design in which we've created essentially 4 distinct treatments of heterogeneity. Now, in order to induce heterogeneity, we're manipulating several variables at a time. We're manipulating in distinct patches. So we've, of course, got a control treatment where soil is untouched. Then we've got a treatment where we've got alternating strips of shallow and untouched deep soil. And so we're creating heterogeneity or distinct patches in soil depth. Then we've got a treatment in which both nitrogen and precipitation are manipulated. So we've got 3 distinct strips or patches of nitrogen, 1 untouched, 1 where we've actually reduced nitrogen by encouraging the growth of microbial species that consume nitrogen using carbon, sawdust, and regular sugar additions. And then we've also got a treatment of enriched nitrogen. And then within this heterogeneity treatment, we've also manipulated the amount of precipitation that's available by adding these rainout shelters, which block about 60% of precipitation on half of the plot. And then finally, we've got a treatment manipulating both nitrogen and soil depth in distinct patches. So again, we've got alternating strips of reduced and regular deep soil. And then we've got 3 strips, 1 in which nitrogen is reduced, 1 in which nitrogen is enriched, and 1 in which nitrogen is untouched. And so we reproduce those 4 treatments so that we've got 4 of each of the 4 treatments and 16 plots total. And this also gives us the ability to look at those subplot level manipulations individually. So we can look at nitrogen individually, depth individually, and precipitation individually as well. And in terms of phenology, we went out for 1 growing season, the growing season of 2021, and we did a weekly phenology survey. So we were checking up on the species in our plots, how far along in their life cycles they were, whether they were in the elongation stage or whether they were starting to reproduce. So the boot stage is the first stage of reproduction. Then you've got the first spikelet and emerged spikelet stages, which are sort of the flowering stages, and then the anthesis stage. And then we use some statistical mixed model tests to see how heterogeneity treatment, these heterogeneity blocks affected the timing of big bluestem life stages. We did check variation as well, but that did not have any effect. So that's a bit outdated. And here's what we found. So let me walk you through this figure. So just as a reminder, the question we're first trying to answer is, how does spatial heterogeneity of soil resources affect the phenology of big bluestem? And once again, we've got 4 different plots, heterogeneity plots. And so we are comparing these plots to one another. And what we're comparing is the timing of development. So on the x-axis here, we've got timing in terms of Julian days, which is just the cumulative day number of the year. And then we've got dates on the top to orient you. And then each of these rows here represents a stage of the big bluestem life cycle. And each of the points represents a different treatment. And so you can compare within each life cycle how the treatments differed. And these treatments where I've got letters printed on them had statistical differences between the treatments. So we see that spatial heterogeneity of soil resources did affect the phenology of big bluestem at 3 life stages, the boot, first spikelet, and emerged spikelet stages, which are all reproductive stages of big bluestem. And if we zoom in here a little bit, you can see that in each of these 3 cases, the treatment that caused big bluestem to enter the stage earliest, which caused an advance in this reproduction, was the nutrient and drought heterogeneity treatment. And you see that this is true at all 3 of these different life stages. So that was kind of interesting. There was no effect on overall flowering length, but there was an advancing effect of this nutrient and drought heterogeneity treatment as compared to other heterogeneity treatments. So then we took a closer look at that nutrient and drought heterogeneity treatment, and we went to the subplot level. If you recall, we compared the different nitrogen treatments to one another, and we stuck within plots to compare nitrogen treatments. So we did this both in the nutrient and depth plots and in the nutrient and drought plots. And the latter is where we saw the significant effect of heterogeneity. And so in this lens, we're asking, how do global change drivers, nitrogen and drought, affect the phenology of big bluestem? And here specifically, we're looking at nitrogen. And what we saw was an interestingly consistent effect of nitrogen. So once again, the letters indicate significance, and we're seeing this effect from the boot through the anthesis stage. So every reproductive stage of big bluestem's life cycle was being affected by this nitrogen manipulation. And consistently across all of these stages, if you look at the filled in black dots, which indicate the enriched nitrogen treatment, you can see that it's separating out from these other treatments as the growing season goes on. So what you see is that enriched nitrogen is causing big bluestem to reproduce earlier, and that this effect is, interestingly, it's cumulative throughout the growing season. So where this difference is smaller in this first earliest boot stage, it becomes larger. The enriched nitrogen treatment becomes more separated from the others as the growing season continues, and the species gets farther along in its life cycle. And interestingly, we saw the same thing in the nutrient and depth and the nutrient and drought plot. Once again, you see these filled in dots separating and getting farther from the other dots as the growing season continues. In terms of drought, we did not see an effect of precipitation treatment on the timing of the bluestem life cycle, but we did see that inducing drought expanded overall flowering length. So it caused big bluestem to flower for a longer period of time. So overall, spatial heterogeneity produced by nutrient and drought manipulation advanced the mean start of the boot, first spikelet, and emerged spikelet stages compared to control. And we saw that nitrogen enrichment accelerated the development of big bluestem across all treatments where we were manipulating nitrogen, and from the boot all the way to anthesis stage. And we also checked to see if nitrogen and drought had an interactive effect, because we were curious if immobilizing the amount of nitrogen available by reducing the amount of water would have this compounded or mitigating effect, but we did not see any effect at all, interestingly, or any interactive effect there. So future directions of my research and other research, we would love to investigate the phenology of other prairie species, because it would be really interesting to continue to study the dynamics of how the phenology of and the resource use through big bluestem's development might later affect the other species it's developing in tandem with, or might affect the prairie from year to year. And I'd like to thank all the awesome members of my lab who helped me out with this study, and all the amazing organizations who helped with my study, and the indigenous people who stewarded the lands that the study took place on for thousands of years before I arrived. And I would love to take any questions you might have.
Thank you, Anna. That was very interesting. I have a question to start it off with. Do they, at your study area, what is their fire regime that they have there? Is it every year, or?
Yeah, so our plot specifically, we burn annually, yes. And then it differs in the rest of Kanza. There's different fire experiments actually going on there.
Okay. And then also, does it, I just don't know enough about Kanza as much as I should. Does it get grazed ever?
Yeah, that's the other major experiment at Kanza. But our experiment does not get grazed. But we do have what are called like the watershed treatments at Kanza, which we've got these patches of grazing and fire going on, which are very interesting.
Okay. All right. Does anyone else have any questions? Feel free to unmute and go ahead and ask. Okay. Well, so I do think that it would be very interesting to see this if you were looking at one of the flowering forbs to see if it has a similar effect, especially some of the forbs that have the much larger flowers. And I don't know, I think that would be really interesting to take a look at.
Yeah. Yeah, I think so too, especially maybe the nitrogen fixing forbs. I'm curious if the rate at which they fix nitrogen changes through their development or anything like that. Yeah.
Okay, last call, everybody. Any questions? Okay, well, thank you very much, Anna, for giving your presentation. So now you're working in California right now?
Yeah, just for the moment. I finished school, so I moved back home for a little bit. But I'm still working on this prairie research actually, and hoping to continue to study grasslands.
Nice. Okay. Yep. Stan just asked a question. Oh, we've got a couple. Stan says, "How are bison at Kanza?"
Yeah. We do. That's the grazing experiment, part of the grazing experiment that's going on at Kanza. There is a herd of bison that's been introduced in the last, I think, couple decades, if not more recently. And they've seen some really interesting results of how bison affect plant species dynamics. Bison graze sort of, they've got taste. Basically, they like certain plants more than others. So it's some really interesting research. I can't recall off the top of my head what exactly they found. I think that they found that bison can reduce the dominance of certain species, but it's a very interesting experiment. I'd recommend looking into it.
Yeah, I feel like I've heard that they've kind of found with grazing in general that it keeps some of the grasses at bay and allows other species, especially forbs, to get more sunlight and thrive just a little bit more. And also what I've heard too is that bison graze much differently than cattle and then goats and the rest of that stuff. So your grazer is very important, which species you use. So Anna, we have another question. Jenny Kissel would like to know, what future research are you doing?
That's a cool question. Well, I'm currently applying to PhD programs, so I'm proposing some research projects. So one thing that I didn't talk about in this presentation that's really quite fascinating about big bluestem and phenology is, if you remember when I was talking about the trade-offs between reproduction and growth, vegetative growth. So big bluestem is a perennial grass. And so that means it comes back year to year. And the way it comes back year to year is because it's got these really well-established roots. And these roots are also a mechanism of spread. So it can reproduce itself using these roots, by producing what are called asexual underground buds. And essentially, these buds contribute a lot to big bluestem's ability to bounce back after a disturbance like fire or grazing. So that's part of the reason it's so dominant, is because it's so good at spreading that way. But essentially, when big bluestem is allocating resources to this vegetative growth, it's producing these asexual below ground buds. But as soon as it starts flowering, as soon as it enters that boot stage, it turns off the flow of resources to that pipeline, and it just pours all of its resources into producing seeds and to sexual reproduction. So because it's reproducing earlier, it's possible that it's producing less of these more disturbance resilient buds. And what we also see in our experimental restoration is this reduction of the dominance of big bluestem in these nitrogen enriched patches. And so I'm curious if perhaps phenology could either contribute to this finding or explain this finding, essentially. If perhaps because nitrogen is causing big bluestem to flower earlier, it is then less resilient to this annual disturbance of fire that we have. And so that's the future research I'm really interested in. I'd really love to study phenology and try to pick apart this relationship between phenology, rates of resource use through time, and how the resource quantities fluctuate throughout the growing season and between growing seasons as well. It's kind of a long-winded answer, but it's just that we don't quite understand too much about how, plants are using resources through time, and how that affects the other plants that are around them. So,
right.
Yeah.
That's interesting. Yeah. I mean, honestly, we for the longest time, for agriculture at least, it's just been you apply nitrogen and other fertilizers at a certain time of year, and we don't really know for the prairie certain plants like when exactly they're utilizing the most of it, all of that kind of stuff. Oh, yeah. Okay. Let's see. Okay, so Jenny has a follow-up question. And since you have a paleobotany background, are there any interesting fossil records related to prairies?
That's a really cool question. I don't know. I wish I knew. When I was doing paleobotany, I was more focused on like cypress plants, but I do know that they've found there's some interesting like ethnobotany, paleobotany overlap where maybe you know this, but a lot of the major agricultural plants, corn and wheat, for example, are grasses that humans have domesticated. And so they found some ancient seeds, I think corn. And that's, it's kind of interesting to study how those seeds have changed through time, but that might be a fun thing to look into. I'm sure there's tons of interesting fossil records. I just don't know about them, sadly.
Okay. All right. So I'm going to, is it all right if I stop your sharing? I have a slide for everybody. If everyone could pull, that one, yes. If everyone could please, oh, hold on, let me get to it. All right. I'm trying to, there we go. All right. If everyone could please help us out and do this little survey. I know you all love surveys, but it really helps us out. And again, thank you very much, Anna, for taking time out of your busy day to talk to us. And yeah, very interesting. I know that the prairie is near and dear to many of our hearts, and learning more about it is always very interesting. So thank you very much.
Thank you all. I love to talk about the prairie, so it was super exciting to get to talk to you all.
Okay, so I'm going to stay on. Everybody else, you can do the survey if you'd like. And thank you very much. And I will see everybody later.